Thank you to Claudia. Congratulations on a great IPO, a NASDAQ IPO end of last year for TiGenix, and welcome onto the ReNeuron board. Very happy to have you. And what TiGenix is doing, which is doing proper controlled studies with stem cells, that is where we all need to get to. So we'll try to follow in your footsteps. Here's a disclaimer, and then moving on. Quick highlights for ReNeuron is that we have a very broad pipeline. The indications are high-value indications. We've got a strong management team in place to deliver on that, and we got the financial backing as well to deliver on that. I'm going to save time, so I don't see a big clock ticking anywhere, but I'm sure somebody will throw something at me. Here's the pipeline. So CTX is in stroke disability. We, in 2017, completed our phase 2, single-arm phase 2A study. The second product is human retinal progenitor cells. In 2017, we completed our single-arm phase 1 safety study. So that's in the box. What's important is to look forward. So next year, we will be in controlled clinical trials, 3 different ones. One for CTX and stroke disability will be a controlled clinical trial in the US. For HRPC, the retinal cells, we will be in 2 phase 2 studies in the US, both of them as well. One for retinitis pigmentosa and one for cone rod dystrophy. So we're very much moving from single-arm studies, mainly based in the UK, to controlled studies in the US. This is where we are in terms of investors and funding. Woodford Investment Management is our largest investor. Wales Life Sciences Fund and Invesco, the second 2. So we have very strong backing from large investors. The cash we had on hand at the last reporting, which was end of March, is £53 million. So we're well funded, but we do have very ambitious programs as well going forward. I often get a question on what the market potential is for the different indications. I don't like to estimate that, but there are luckily analysts that do that. So there are 3 different financial analysts that are covering us, and this is the range of estimates that they have. So CTX for stroke disability in a population 6 to 12 months post-stroke. If you assume that a certain very low market penetration, I think quite a low price they've used as well, but $40,000 to $60,000 for a single injection. It's a one-time treatment. The peak sales comes between $1 and $4 billion. That's a wide range, but I'm happy anywhere really in that range is fine. And the retinal program, it's a smaller indication, but again, we come here to peak estimate sales between $500 million and $2 billion. So I think what it is an illustration that there is a large market potential if and when we are successful. So let's dive into the CTX cell product a little bit. It's cryopreserved, and you can see Randolph Courtling here, head of research, picking a vial out of the freezer, probably about minus 180, so it's smart to have thick gloves on when you play around with that. Because it is cryopreserved, that means that we have a proper shelf life. We have a 6-month shelf life on these cells right now. That means you can ship them to clinical trial sites, store them there, and use them when you need. And this time next year, will probably be about 12-month shelf life on that, so something that we build out. That makes a big difference from phase 1 when the shelf life was 8 hours. So you can imagine how it is to do clinical trials then. It was possible as well, but it's usually done a single site, very close to your manufacturing. The stroke indication is not a popular one. A lot of companies have tried to come out with new drugs in acute stroke, and they failed. Acute stroke is a difficult area to show that your drug works because people do get better very fast, just naturally. So you're fighting against a placebo group that are improving. What most people haven't done is to look at patients who are completely stable, so months or even years after stroke, when all the natural recovery is finished and they're left with some disabilities. Typical, the disabilities have to do with mobility, and they end up being in care homes or in a wheelchair, and that's where we come in. There are no pharmaceutical treatment in this group. In the acute side, there is TPA from Genentech. That's the only thing that's available. But in the chronic side, there's absolutely nothing. We did a phase 1 study, dose-ranging study that was published in The Lancet a year ago. The top dose from that study was 20 million cells. We inject these cells into the brain, into the putamen, on the affected side where the stroke has happened. So this is a single injection. This is brain surgery, but it's very much bread and butter brain surgery. This is what the surgeons do, neurosurgeons do. The procedure takes about 2 hours. Most of that time is spent setting up the surgery. So they put on a frame, a plastic frame. They set up MRI so they know exactly where they're going to drill a hole. But they do drill a hole, and then they inject the cells through that. And the injection itself takes about 20 minutes because they like to distribute the cells around the affected area. After that, we did a single-arm phase 2 study. The reason for single-arm is that ethically very hard to do placebo surgery unless you have some real evidence of efficacy that is worth the effort. So that's what this phase 2 really has done for us, is to show that it does that. And I'm going to skip through this. This is very much how many patients were treated, 23, age range 40 to 89, median was 7 months post the stroke. So, and obviously we measured all these patients, make sure that they were completely stable, that they were not improving as we treated them. So here are the results. These are 3 months results and 12 months results. The 12-month data were just in hand. There was a press release about a week ago on that. And the most important measure, there are a lot of different ways to measure stroke. The one that FDA likes the most, so that means it's the most important one, is called Modified Ranking Scale. It is a very rough scale, and I'll show you that later. It's a scale of disability and dependency. So can you live alone at home, or do you need help, or do you have to live in an institution? So that's kind of a very, very rough scale, but it's an important scale. So one change on that scale is clinical significant. We had 7 out of our patients of the 3 months, so 30% out of at 3 months that reached that. And at 12 months, we still had 7 patients. There were some dropouts in between, so the percentage changes. But basically, I think what we conclude from that is that the initial response we get after 3 or 6 months, that will last for a long time. And it kind of makes sense as well that, you know, if somebody has had issues, been stuck in a wheelchair, if you're able to get that patient out of the wheelchair, moving around again, maybe with a Zimmer frame, maybe with a cane, they're not going to end up back in a wheelchair unless they've had another stroke or something else has happened. So, you know, whatever change you have is a permanent change. So the conclusions from our PISCES 2 study was it was well tolerated. That's one of the more important things still. We were able to do these surgeries without any major issues. We're very happy with the response on modified ranking. There's one competing company in California called SanBio. They have not seen any modified ranking responses in their studies, so we're very happy to see that we can move the needle that far. Future studies will be focused on subgroup of patients who have even better response, and you can see that on the next slide. Because that's PISCES 3, that's our next study. It's going to be a controlled study. It's a phase 2b study. We do not believe we will get approval on that study, although with RMAT designation, etc., maybe. But I think from a public expectation, you should expect another study to happen after this, being done either by us or someone or a partner. Primary endpoint will be modified ranking 6 months after treatment, and we will include patients who have it's called NIHSS4 on the upper arm movement, so that we only will include patients who have some movement in their upper arm so that they have some chance to improve. That's one of the learnings from phase 2, that if the patients do not have any upper arm movement, very hard to move them down the scale. All the patients will be modified ranking 3 and 4, and that means so this is a good illustration of what it means to move from one of these modified ranking scale down to another. This is based on a very large group of patients, over 40,000 patients in Sweden, in cost of disability. As you can see here, so if someone modified ranking 5, that's someone who's bedridden. They cannot even lift the head off the pillow. Okay, obviously very expensive to care for, over $100,000. Someone in modified ranking 4, they're in a wheelchair. Cost drops a little bit, but it's very expensive still to care for. And we're not including family here. This is spending outside that. And if you can get someone from modified ranking 4 to 3, that's a huge drop in care cost, and obviously an enormous benefit to the patient. 3 really means that you can start living at home. You still need some help. If you can get them down to 2, they can actually live at home without any help from any people to come in every day. So, this is our goal. We're getting the 3s and 4s into our next study, and the goal is to get them down 1 level to 2s and 3s. How many minutes did I have left, did we say? 2? I'll jump very fast, 4 minutes, thank you, through the human retinal progenitor cell program. The retina is a wonderful place to do cell therapy. Your eye does not generate new photoreceptors. You're born with all that you ever have. So if they're lost due to disease, they're not coming back. So cell therapy is a great potential to actually do something here. This is a program that we actually licensed in the IP from Skepin's Eye Research Institute at Harvard. We had the cells, they had the IP, so we kind of had to get together. And we worked really well together for a number of years on this. Retinitis pigmentosa was presented 2 presentations ago, so if you remember, and I won't go through it again. We're luckily a bit further ahead than you guys. So we're in the clinic. We just finished the phase 1 portion of our clinical trial. It is at Massachusetts Eye and Ear Infirmary in Boston. It was a dose escalation study. The top dose was 1 million cells. These cells are injected into the back of the eye, as you can see in that illustration. I do have a video as well, but I'm sure you don't want to see it. Because it's not something that you want to see too much of, I think, for most of us. But what's so impressive with these surgeons is that they have such steady hands. It's unbelievable. So what they actually do is to inject the cell into the back of the eye, and you go in, you detach the retina temporarily by a little air bubble, and then you put the stem cells into that little bleb, and that spreads across the retina. And it will stay there. So in animal studies, we see 80% of those cells are still there after 12 weeks. Can't really do that in humans. So the phase 1 is done. What we're doing now in phase 2A is that we're moving up the level of vision. So the people in phase 1 have very little vision left, and their retinas are pretty damaged. So you're not going to see any efficacy in them. But obviously in the beginning, first do no harm. Now in phase 2A, what we're doing is we're going to patient and people at the same time. We're going to patients who have more vision left. So in phase 1, we're talking hand movement or light perception. That's the vision level. And then you start moving into 2800, 2400, etc. As we go forward. When that's done, we will go into a controlled phase 2B study in the US. There's a second indication called cone rod dystrophy. It's a smaller indication. We do not need to do a phase 1 on that indication. We can jump straight into a controlled phase 2, and that's what we will do as soon as the RP program is up and running. There's a very, very sexy program in research called Exosomes, and you might start hearing a lot about exosomes out there when you're talking to friends and colleagues. I'm not going to talk much about it because the time doesn't really allow for it, but it's a very, very interesting program. Exosomes is the way cells talk to each other, and it could be a big part of the mechanism of action of any stem cell, and it's kind of how we started with exosomes when we're looking at mechanism of action of our CTX cells. It is a very hot field, so we're looking at doing collaborations here. Oncology is the indication that we're looking at first of all. What we're lucky with here is that these cells, these exosomes, are made from the manufacturing of our CTX cells. So we are already in GMP lines, so we have kind of a head start for most other companies when it comes to exosomes. A lot of this is grant-funded, and I think you'll hear a lot more about exosome from a lot of other companies over the next few years. I will leave you with the near-time timelines, but very much what I said. This year, completing a phase 1 study for retina and a phase 2 study for CTX and stroke disability. Next year, we will be in 3 randomized clinical trials. All of them will be 100% done in the US. Thank you very much.